Welcome back to the Deep Dive. Today, we are charting some really new territory in biology.
Yeah.
We're talking about how a cell's physical address, its microenvironment, basically dictates its entire job description.
Yeah, exactly. It's a process called cellular regional specification.
And that's this idea that the neighborhood a cell lives in determines its function, its development, and, crucially, its role in diseases like cancer or Alzheimer's.
For years, researchers have tried to map these neighborhoods using something called spatial transcriptomics, or ST, which measures all the RNA in a piece of tissue while keeping that location information intact. But we have always had to face this one really frustrating trade-off.
It's a classic scientific dilemma, right? Think of it like trying to map a huge city.
Right. So option one is like the comprehensive census. You get a complete, unbiased reading of everything happening, the whole transcriptome.
But the map is blurry.
Exactly. You can only capture data in these large city blocks, which scientists call spots, and each spot often contains dozens of different cells. So you lose all the detail of the individual houses, and you miss everything happening in the alleys between the blocks.
So that's what's called sequencing-based ST, something like Visium.
Yep. And then you have option two,
which would be the sharp single-house resolution map. It's crystal clear. It shows you every single cell.
But you only get to track a pre-selected group of residents, a tiny panel of, say, 500 genes or so.
Yeah.
You capture beautiful spatial detail, but you completely miss the whole complex story.
And that's image-based ST, like Xenium. Great for checking something you already know, but, well, terrible for discovering something new.
So the Holy Grail has always been a map that provides both single-cell resolution and whole transcriptome coverage, and across the entire piece of tissue.
And that, listeners, is the mission this deep dive is built around. We are diving into Spotiphy, which is a new computational method that aims to bridge this exact gap.
The key is that Spotiphy doesn't use new lab hardware. It uses really smart software, a generative model, to integrate 3 different types of data,
turning those blurry bulk measurements into a high-definition, cell-by-cell molecular map.
We're going to walk you through exactly how this algorithm performs that magic, and I think more importantly, the profound new biological secrets it has already revealed in models of Alzheimer's disease and in human breast cancer tissue.
Okay, let's get into the engine room. How does Spotiphy actually pull this off? How does it achieve single-cell spatial whole transcriptomics?
It all comes down to a probabilistic generative model.
Okay, so what is that in simple terms?
It's basically a powerful statistical engine. It's designed to infer the most likely true state of something, even when it only has incomplete, noisy data to work with.
And it needs 3 ingredients to do this.
It does. The first is that low-resolution foundation, the sequencing-based ST data we talked about, the blurry city blocks. This gives the model the bulk gene counts for defined areas.
And the second input is the intelligence, I guess you could call it, the single-cell RNA sequencing data.
Right, the reference library. Spotiphy first analyzes this library to identify the unique fingerprint of every cell type, what genes define an astrocyte versus, say, a neuron. It uses some pretty robust stats to create this signature matrix, making sure it's accurate.
And the third piece, which sounds critical, is the visual guide, the high-resolution histological images, the H&E slides.
This is a huge step. The algorithm analyzes this image to perform what's called nucleus segmentation.
Meaning it finds the exact location and counts the number of individual cell nuclei inside every one of those blurry ST spots.
Exactly, and that's the crucial step that lets you translate bulk gene numbers into discrete cellular units.
So once it has those 3 inputs, the bulk genes, the cell fingerprints, and the cell count per spot, the model gets to work.
And it does 2 things at the same time within each spot. First, deconvolution. Based on the spot's total gene expression and the known cell signatures, the model figures out the cell type proportion.
So it might conclude, okay, this spot is 60% glial cells and 40% neurons.
Precisely. But here's the real leap, and what the researchers call the decomposition step. Spotiphy doesn't just stop at proportions.
It goes deeper.
It uses Bayesian statistics to partition the total gene counts and assign them to the individual cells it has estimated are within that spot. This generates what they call inferred single-cell RNA data, or is-EC-RNA.
Wow. So that's how you go from blurry bulk data to whole transcriptome information for every single cell.
That's the magic. And then they tackle the other major flaw of these platforms, the missing data in the outspot space, the alleys between the blocks.
The non-capture areas.
Right. Since up to half the tissue can be missed by the physical spots, Spotiphy uses something called Gaussian processes, which are really just sophisticated interpolation tools, to reliably estimate the cell types and RNA data for the cells in those gaps.
It just fills in the blanks based on the patterns it sees nearby.
Seamlessly.
So, okay, let's recap this first part. The goal was to overcome that frustrating trade-off between seeing everything and seeing clearly.
Right. Resolution versus coverage.
And Spotiphy does it by combining 3 data types: the bulk sequencing, the single-cell reference, and the histology image.
Yeah.
And the key outputs are, 1, figuring out who is in the spot,
Deconvolution.
and 2, assigning whole gene expression data to every single one of those cells.
Decomposition and the RNA, plus the imputation for the gaps.
Perfect. So before we jump into the amazing biological results, here's a question for you to think about. Spotiphy seems to handle rare immune cells with incredible accuracy. Why do you think other, older methods, which maybe rely more on just pure math, traditionally struggle so much to accurately place these rare populations?
That's a great question, and it's a perfect setup for what comes next. A lot of older methods are just, well, they're overly sensitive to the quality of the reference data.
So if your library of cell fingerprints is sparse, maybe it only has a few examples of a rare cell, the other algorithms just can't figure it out.
They struggle to extrapolate. But Spotiphy's approach of actually modeling the gene counts for each cell seems to make its predictions way more stable. And the benchmarks they ran confirmed this.
Which brings us to section 2. The biological payoff. What did they find in the brain?
So they compared Spotiphy against 13 other methods and showed it consistently produced the highest correlation coefficient with the ground truth data.
The ground truth being data from the high-resolution image-based platforms.
Exactly. Plus, it was substantially faster to run, which, when you're processing these massive data sets, matters a lot.
Okay, let's talk about the Alzheimer's mouse brain model.
They used a really detailed reference with 27 cell types. And Spotiphy, it not only delineated the major structures, like accurately mapping the cortical layers, but it really excelled where its competitors failed, finding rare cells.
Like neutrophils.
Like neutrophils. These are transient immune cells, usually pretty scarce in a healthy brain. Spotiphy precisely predicted an enrichment of these neutrophils concentrated right around the ventricle.
And that's a key area for fluid exchange in the brain.
It is. And they confirmed this prediction using standard lab techniques. So this wasn't just a guess. It was an accurate, high-res location of a cell population that competitor methods often misclassified as something else entirely, like macrophages.
That is just critical validation. If you can't accurately locate the rare immune cells involved at the start of a disease, you can't possibly hope to target them effectively.
Exactly. But I think the most important biological discovery unlocked by Spotiphy's inferred data was the regional specification of astrocytes.
Astrocytes, the star-shaped support cells, usually people just treat them as one big group, right?
They do. But Spotiphy revealed they have distinct jobs based purely on their location.
So their neighborhood actually changes their function. What were the differences?
The analysis showed that astrocytes in the cerebral cortex, or CTX, were highly active in pathways related to neuronal differentiation and cell fate. They were focused on helping new neurons figure out what they're supposed to be.
Okay. And what about the others?
Meanwhile, the astrocytes in the thalamus, which is a deeper, older brain structure, were focused on neuronal synaptic plasticity fine-tuning existing connections.
So it's like discovering the maintenance crew in City Hall does completely different work than the crew in the suburbs, even though they have the same job title.
That's a perfect analogy. Their environment dictates their specialized activity.
This has to change how we think about brain disease. If we see astrocyte problems in Alzheimer's, and we now know that problem is functionally different in the cortex versus the thalamus, our drugs have to reflect that.
That's it. A drug targeting plasticity might completely miss the differentiation problem happening just a few millimeters away. It forces us to ask, is this disease one uniform thing, or is it a collection of specialized regional breakdowns?
Okay, that brings us to our second recap. We've seen the tool accurately map rare immune cells and, maybe more importantly, reveal the functional specialization of support cells in the brain.
And the implications for drug design are just huge. It forces researchers to think about location, not just cell type. Which leads to this question. How could this kind of regional functional map change the way we develop drugs for neurological diseases?
Let's continue that exact theme in section 3. Microglia, cancer, and the tumor microenvironment.
So microglia, they're the brain's resident immune cells, and they're crucial in Alzheimer's. Spotiphy analyzed them and found 5 subclusters that aligned perfectly with histological regions, like the striatum and the cortex hippocampus, or CNH.
And when you focused on the disease-affected cells?
The microglia in those CNH and thalamus regions were almost all from the AD sample, and the analysis showed they matched the markers for disease-associated microglia, or DAMs.
The cells that activate in response to amyloid plaques.
Right. And this was confirmed by actually staining for the beta amyloid protein and finding high signals in those exact same regions. It all lined up.
And let me guess, even within the DAMs, there was regional specialization.
You got it. The DAMs in the cortex and hippocampus region showed increased upregulation of immune response genes. They were more highly activated, more inflamed than the DAMs found in the thalamus. So even inflammation itself isn't uniform.
That's phenomenal. Okay, let's shift from the mouse brain to human disease, breast cancer. The tumor microenvironment, the TME, is this notorious mix of tumor cells, immune cells, all kinds of structures.
And this is where the ability to generate that inferred single-cell whole transcriptome data was so key. It allowed them to perform what's called infer-CNV analysis.
Which is inferred copy number variation.
Basically, it looks for big structural changes in the genome, which cancer cells often have. And Spotiphy could do this at a single-cell level, successfully differentiating the malignant tumor cells from the normal ones.
And this high resolution allowed them to map the actual physical organization of the tumor.
For one sample, yes. The tumor cells formed 3 distinct spatial domains: an outer layer, a middle layer, and an inner layer. And as you'd expect, the number of immune cells generally decreased as you move toward the center.
Fewer immune cells deeper in the tumor. That makes sense.
But the surprise came when they used a tool called cell chat to analyze the communication between the cells. You would think the strongest immune reaction, the most aggressive crosstalk, would be where the most immune cells were, in the outer layer.
Wait, so the highest density of immune cells was on the outside, but the strongest reaction was in the middle layer. That's totally counterintuitive.
It is. Spotiphy showed the strongest immune reactions were happening in that ML domain. This suggests it's not just about the quantity of cells, it's the quality and intensity of the communication that really matters.
Did they have an explanation for that?
The analysis offered one. The ML domain showed an upregulation of markers associated with cell mobility, things involving the collagen family. It suggests this middle layer is a dynamic battleground, an active zone of infiltration and aggressive communication, not just some passive buffer.
And just to close the loop on the tech side, this all validates that imputation step, right?
Right.
Filling in the gaps.
Absolutely. The data Spotiphy imputed for those out-of-spot areas was actually more accurate than what some competitor methods achieved for the cells they successfully captured in their spots.
The computational model is outperforming the physical hardware.
In some cases, yes. Which leads us directly to the future.
And that really feels like the path forward for biology, doesn't it? Using smart computation to get around the physical limits of our hardware. Which brings us to Section 4, future trajectories.
The main value proposition is just, you eliminate that choice. You don't have to pick between resolution and coverage anymore.
And this approach isn't just for Visium data.
No, not at all. Spotiphy's imputation function could be used to rescue data from other platforms like slide tags, which can suffer up to 75% cell loss during the process. You could use the model to recover that lost data, which would be a huge boost to statistical power.
The creators are also looking at integrating more advanced concepts. They mentioned deep learning vision transformers to better correlate cell types with what they actually look like in the image.
That would make the cell assignment even more robust. And imagine applying this not just to a flat slice of tissue, but in 3D.
Building a full 3D model of the tissue.
By leveraging the similarities between adjacent slices, they aim to reconstruct the full 3D tissue structure, building a complete molecular anatomy without needing incredibly expensive 3D imaging hardware.
And finally, they're planning to integrate multimodal data.
Right. Think about layering this whole transcriptome map with functional data, like single-cell ATAC-seq, which maps which parts of the DNA are accessible.
So you'd see not only what genes a cell is expressing and where it is, but how its DNA is being regulated in that specific spot.
It would be the most comprehensive, spatially resolved view of regulatory mechanisms we've ever had.
Okay, let's unpack all of this. Spotiphy, it really feels like a watershed moment. It turns what was previously low-fidelity, patchy spatial data into, I don't know, molecular GPS maps. It's letting biologists move past just identifying cell types to fundamentally understanding how a cell's environment changes its job description, whether that's fine-tuning plasticity in the thalamus or aggressively interacting with collagen in a tumor boundary. We're moving into the realm of high-definition molecular anatomy.
What's so fascinating to me here is that for decades, we've been forced to view these incredibly complex tissues through very limited lenses, either broad and blurry or narrow and sharp. Spotiphy provides a computational layer that it eliminates that choice.
Right.
It suggests that a lot of these subtle regional variations, like the specialized function of microglia or the layers of immune response in a tumor, that wasn't just experimental noise, it's genuine, profound biology that we simply couldn't detect because our maps weren't good enough.
So final thought to leave everyone with. Given that we've already seen this profound functional specialization in healthy and diseased brain cells, and this highly localized immune activity in human breast tumors, what other tissues, maybe your liver, your lungs, your pancreas, are hiding these kinds of functional distinctions tied only to their precise location that we have yet to discover?